Hello students, a warm welcome to all of you in ITLS Academy. In today's lecture, we will discuss about the Ramachandran plot. In today's lecture, we will understand how we retrieve Ramachandran plot of a particular protein. Okay, to retrieve the Ramachandran plot, we use ProCheck and PdbSum. These are the bioinformatics tools that are used to retrieve the Ramachandran plot of a particular protein. Ramachandran plot. Okay, so in this lecture we will understand how we retrieve the Ramachandran plot from PdbSum. Okay, so we start today's lecture and learn how we retrieve the Ramachandran plot from PdbSum. Okay, to retrieve the Ramachandran plot from PdbSum, we simply search PDB. We simply search PdbSum. Okay, and open this one. Okay, and this is a home page of PdbSum. Okay, here we search our target molecule from PDB code, from AlphaFold model, from text speech, from search by sequence. We also put the UniProt ID, PFAM ID or ENSMBL ID to search our target molecule. Here I search our target molecule by the help of PDB code, and our target molecule is 1HKG, and simply find. Okay, and this is the all information related to our target molecule. This is the information, this is the literature information of our target molecule. This is the enzyme reactions related to, this is the enzymatic reaction of our target molecule. Here the information related to the protein chain. This is the 3D structure of our target molecule. This is the PDB ID, name, title, source. All information related to our target molecule is present here. Okay, and this is the Ramachandran plot of our target molecule. Okay, and this is the Ramachandran plot of our target proteins. Okay, this is the allowed region, this is the partially allowed region, and this is the disallowed regions. This is the information. In x-axis there is a phi bond, and in y-axis there is a psi. This is the summary of Ramachandran plot. Okay, these are the amino acids which are present in allowed region, and disallowed region only 70 amino acids are present. Okay, in this is the non-glycine and non-proline residues are 401, and residues are 2, glycine residues are 32, and proline residues are 42, and total number of amino acids is 457. Okay, and this is the G factor of our target molecules. Okay, and this is the important information related to our target molecules. Okay, and in this way we easily find out the easily retrieve the Ramachandran plot of our target molecule by the help of, whenever we want to search our target molecule by sequence, then we submit the FASTA format of our target sequences. Suppose first we retrieve the protein sequences from NCBI. Here I open first NCBI. This is the first. Okay, then we open, then in all database we select protein and search our target molecule. Here I search hexokinase or search. Okay, then select our target molecule. Okay, then convert this genpeg to FASTA format. Okay, then copy these sequences and paste these sequences in this search bar, and whenever we search our target molecule by the sequence, then we find the 129 proteins. Okay, these are the proteins which have a similarity to our target to given sequences. Okay, and this is a PDB ID. Okay, and this is the model, this is the information related to model, this is the length information, these are the similarity, this is the length of amino acids. Suppose this first have a 49.5% similarity with our target sequences, and this is the Z square value, this is the information related to the ligands, and this is the protein name. In these proteins, in these protein, we select our target molecule. Here I want to search the human glucokinase, so I select this one. Okay, and this is a result of our target molecule. This is the information related to the protein chain, ligands, metal, water molecule, enzymatic reactions, and literature information. Okay, and this is the Ramachandran plot of our target molecule, and this is the information related to the Ramachandran plot. In this way, we retrieve the Ramachandran plot by PDB ID or by sequence. Okay, to retrieve the Ramachandran plot from PDB, first we open PdbSum. Okay, and in PdbSum we search our target molecule by PDB ID, by alpha fold ID, by alpha fold name, by uniprot ID, by searching by sequences. And in this video lecture we understand how we retrieve the Ramachandran plot by the help of PDB ID or by the help of sequence. Whenever we search the sequence or whenever we search by PDB ID, then we simply put the PDB ID of our target molecule and create the Ramachandran plot. Whenever we retrieve the Ramachandran plot by sequence, then we first submit the FASTA sequence, then we retrieve the PdbSum, generate a list of target molecules which have a similarity to our target sequences, and then in this list we select our target molecule and create the Ramachandran plot. In this way we easily create a Ramachandran plot by the help of PdbSum. Okay, and this is all about today's lecture. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share and subscribe ITLS Academy. Thank you.